yeah, it's sad that Vinny can't present this because this is fun work. But I'm very happy to talk about it because we both worked on it, and it's like some new or old thing, but depending on how you look at it, but something that is emerging currently in the cancer field, and that is the somatic evolution of like extra chromosomal DNA in the context of cancer. So as I said, so that's me. But Winnie, I, and Yuri, who was at the time a postdoc at the Max Planck in Plön and now is in Princeton, actually, we worked on this from a theory point of view, and we published this paper in bioRxiv 2019. And then the very last conference that I went to before the pandemic, there were like everyone that worked experimentally and clinically on ecDNA, and there's like people like Paul, Michel, Anton, and Howard. And we decided to team up. So they had a lot of data, and we had theory, and we thought, okay, let's try to bring it together, and like maybe we can make something useful out of it. And so that took us a little bit, but today I can talk a little bit about that. So what is ecDNA? So extra chromosomal DNA. These are ring-like structures, really circular DNA, and we think they form like at moments of like genomic instability, like cell divides. There's genomic instability. Certain parts of chromosomes break, or you lose bits of DNA, and these bits of DNA that you lose, they form these ring-like structures. They have been observed sometimes in the 80s, just by looking under the microscope in leukemias, for example. They were called double minutes. So like you have ring-like structures, and they didn't really separate properly. So you had like two rings being stuck within each other, and you could really see that under the microscope if they were big enough. But now, in the last years, with like modern sequencing technology, and if you really actively look for them, it actually turns out that they are very prevalent across cancers. So it seems like in 33 to maybe even 40% of all cancers, you can detect the presence of these ring-like structures. And what is even more interesting is that very prominent oncogenes, MYC, EGFR, MYC-N, and others, seem to be amplified on these ring-like structures in cells as well. So there seems to be some importance to them. And I'm sorry, these are Winnie's slides, so I have to check a little bit what's going on. But they also, if you look at clinical impact, there are first studies that show. So it's known that chromosomal instability is a bad predictor. So if you have a cancer and you find chromosomal instability, patients do worse. Among the patients that have chromosomal instability and also have ecDNA, they seem to do even worse. So the presence of ecDNA is a worse predictor of outcome than just chromosomal instability itself. So from an evolutionary point of view, why do we think that ecDNA is an interesting problem, and why has it interesting properties? And it has been thought that these ring-like structures, like in bacteria, for example, they start to segregate randomly. Like usually, if you have chromosomes, if a cell divides, what happens is you have like big machinery in your cell that makes sure that like each daughter cell gets the same amount of chromosomes. Of course, sometimes in cancer that goes wrong, but usually that is so. You segregate in diploid genomes. For ecDNA, you don't have, for example, you don't have these centromeres. So there's this mechanism that makes sure that you equally segregate just doesn't exist. So what happens is you just start to segregate them randomly, and that immediately allows you to like produce high copy numbers within single cells. So if you start with a cell that has a certain number of ecDNA copies, you amplify ecDNA during the cell proliferation process, and then you segregate these 2i copies randomly by a binomial distribution. And if you count this process, you immediately have like different possibilities. Like you start with a single cell, this ecDNA gets amplified, and then you have two ecDNA that you can distribute. And then you have two possibilities. You either have two cells with one copy of ecDNA, or you have like one cell, no ecDNA, one cell with two copies of ecDNA. Say we have this copy with the cell with two copies of ecDNA, and we follow that in time. So this divides, and now I have like 3 possible outcomes, right? So I have like one cell with 1 copy, one cell with 3 copies, one cell with 2 copies, or 2 cells with 2 copies, and one cell with no copy, and one cell with 4 copies of ecDNA. So we immediately see there's a lot of randomness involved, and we can produce diversity in this process. So when we started this, we thought, okay, that's a funny process. We want to model that from a theoretical point of view, and then we want to see what are certain predictions that we can come up with under these evolutionary hypotheses, and then can we see these things actually in data. So 3 main things I want to talk about today. So is this assumption of random segregation actually true? Given that I explained it so long, the answer is probably yes. If it's true, what are the patterns of heterogeneity that we actually observe, and can we describe them theoretically, and does this agree with what we see in data? And is it easy or difficult to distinguish, like, if we have these ecDNA elements, do they contribute to cancer growth? Are they under positive selection, or are they neutral? Are they just random byproducts of like chromosome instability, right? So we want to talk about these 3 things. So to show that ecDNA is completely random, Paul, Vinit, and Josh is the PhD student in Paul's lab who actually did the work on the microscope, did a cool experiment. So what they did is they took daughter cells, stained for these ecDNA copies, and then let a cell divide, and then take these 2 daughter cells after division and basically counted the fish. Lights off. Let me try that.
Yeah, we'll try. I know. Oh. Do that one. Well, you know, I keep, you know, doing like trial and error methods.
So what they did is it a little bit better? Or? What they did is they stained these copies of ecDNA in these cells. They let these cells divide, and then they took these 2 daughter cells after division and basically counted how many ecDNA is in each daughter cell. And if you do that for sufficiently many cells, you can just come up with a very simple theoretical prediction, right? If segregation is always equal, and you ask what is the proportion of ecDNA segregated in each daughter cell, if it's always equal, it's like this sharp line here at 0.5. If it's completely random with a probability of 0.5, you get this distribution of fractions that is centered around 0.5. And it also could be that they are random, but there's a bias, like one cell always gets a little bit more than the other cell. And then you would get like these distributions that have, they are wide, but they have like 2 peaks, one at like 0.3 and 0.7, for example, if these are these binomial probabilities, right? And if you do that in the data, so these are different cell lines, different oncogenes amplified on these cell lines. For example, here's the MYC gene, the FGFR gene, the CDK4 gene, MYC-M, and so on. So different oncogenes amplified on ecDNA in different cell lines. You do this counting by fish. You always find this very nice distributions, wide, centered around 0.5, really suggesting completely random segregation. So that's nice. So random segregation seems to be true. What can we say about the population dynamics and the diversity? So the first thing we can ask is a very simple question in a sense. What is the evolution of fractions of ecDNA copies within the population? So we ask, I start with a single cell, with a single copy, then you have this proliferation process, cells divide, and then what I want to count is the numbers of cells that don't carry any copies of ecDNA and the numbers of cells that do carry copies of ecDNA, right? And then we can do stochastic models. So you see the single lines here. There are stochastic simulations of exactly this process in an exponentially growing population. And these black lines are cells with ecDNA. These are cells without ecDNA, and these are the cells with ecDNA in the stochastic simulations. And then you can also do some analytical calculations, and these are these dashed lines. And what you observe is a dynamics that looks like this. So you have 2 lines. So basically, I'm very sorry. So the PDF, there are some equations and some pictures missing here, which is very bad. But the important information is here. So you can write equations here. So n minus is the numbers of cells that do not carry ecDNA, and you would have a similar equation for n plus that looks very similar, it's very symmetric. And what happens is the fraction looks like this. So if ecDNA is neutral, so they are there, but they don't contribute, they don't give a fitness advantage. What happens is that over time, the fraction of cells without ecDNA is growing. And like if you would run this like to very, very large population sizes, you would almost reach a fraction of 1. And the reason is very simple. If you lose ecDNA, that's an absorbing state for your system because you don't gain it back, right? So you have the cells that always distribute copies of ecDNA, but once you lost it, it's lost. You don't get it back. So these cells start to accumulate over time and they start to dominate these tumor populations. The situation is different if ecDNA is under positive selection. So you see this funny peak here. So first, the fraction of cells without ecDNA on average is increasing, but in the long run, the tumor starts to get dominated by cells actually carrying copies of ecDNA. So again, if you run this to very large simulations, you basically go to fractions to 1. So yeah. So it in theory, it's possible. Like we are discussing this. This model is the simplest model where we just exclude that, but there are hints, or like you can integrate or reintegrate like copies of ecDNA into the chromosomes that then under certain conditions jump out. I mean, it's not super well established, so it's not like that's for certain, but it seems like I'm not talking really about treatment here, but if you give treatment, it seems what happens is sometimes copies of ecDNA integrate into chromosomes. And then if you take treatment away, they kind of jump out of chromosomes again. So it sounds magical, but so Paul, for example, believes that this is really happening. So in that sense, for example, you could reintroduce ecDNA. If you think of introducing ecDNA by a random process of like breaking your chromosomes and then making a new copy of ecDNA, that's possible, but it will be a completely different copy. Like producing, we are talking here about oncogenes where you have like, there are millions of base pairs large, they're really big. And you have an EGFR oncogene amplified of ecDNA plus some epigenetic regulator and stuff like that. So they're really complex things. So producing this by chance a second time in the tumor, that's extremely unlikely. But this integration, reintegration, that is possible. So again, under neutrality, you lose ecDNA kind of in your tumor. Under positive selection, you start to get dominated by ecDNA. And if we check in both patient data and in cell line experiments, so this is each dot here is either one patient or one cell line. So these are glioblastoma patients, for example, where EGFR is amplified on these ecDNA copies. You see these fractions are in the order of like 0.8, 0.9, something like that. So high fractions of cells carrying ecDNA copies. If you have cell lines, this is McN, so this is neuroblastoma. So this is, again, fractions very, very high, and then cell lines fractions even higher. Under this very simple measure, already suggesting some positive selection for these copies of ecDNA. Second quantity we can look at is how does the mean copy number of ecDNA change over time. So we go from like a deterministic description of the system where we just look at fractions to a stochastic description of the system, but first look at the moments. And what we find is that under neutrality, the average copy number of ecDNA per cell stays constant over time, which is funny. I told you that the fraction of ecDNA goes down, but the total number averaged over the total population stays constant. So there's like, you get a very small subpopulation that carries a lot of ecDNA, but overall that is averaged out over the total population. Whereas under positive selection, ecDNA copy number is slowly increasing over time. So with time, the longer you wait, the higher the average copy number of ecDNA in your tumors. And the same is true for if you look at second and third moments, so the variance is increasing even faster, so you get a lot of variation. And there's a slow increase in variance if you have a neutral process. And we can test this experimentally. So Howard did a very nice experiment where they did this. So they had a haploid cell line, and they used CRISPR-Cas to produce ecDNA elements in this cell line, and then you can follow the fraction of, the mean copy number of ecDNA in this cell line over time. And there's an initial phase here where so this is like a negative selection phase where you lose copies of ecDNA simply by cells don't like being CRISPR-Cas. So there's a lot of cells dying here. But then you reach a constant fraction here. So these are the cells that you kind of artificially selected in this experiment that survive this procedure, that still carry ecDNA. And if you then follow these cells over time, ecDNA levels stay constant, which is fine because we CRISPR-Cas a completely random part of the genome where we would not expect any positive selection. Manix? So this one, yes. The slides I showed you before, these are patient data and cell line data with like oncogenes amplified, yes. Right. Yeah, so that's a detail that I completely skipped over, but you picked up on it. So there are cell lines that are historically isolated from patients, or glioblastoma cell lines mainly, but they're also like a colon cell line and I don't know what else is there, but like different oncogenes amplified. And the reason they have these small differences in shape is because the actual width of the distribution, like how wide it is, depends on the overall average copy number that you have. Because in some of these cell lines, the copy number is extremely high. They have like on average 200 copies of ecDNA per cell. It's really a lot. And you can imagine, like, if you segregate that, you get a slightly wider distribution than when you segregate like a cell line experiment where the average number of ecDNA copies is like 50 or 40. It's just, it's not really underlying biology. This is simply like when you segregate after the single cell line experiments, and you segregate is just a game of numbers, like how many ecDNA copies you can distribute like in each side. So. Yeah, of course. So if you look here, so this is neutral selection, right? Negative selection, like you have the same line but much steeper. And if you look here, like you basically have like a decline of like ecDNA copies. Yeah, that's a good question. I'm not sure. That's a good question. So this one is quite extreme, I think, but it's really because like the way you design the experiment, I think. Right. We probably could. We were not. To be honest, we were not really interested in like this negative selection phase. We wanted to show that like this neutral. But you probably could. You always need assumptions then on like cell proliferation time and stuff like that, because the number of cell divisions matter for like how you distribute them and like how fast they decline and so on. So, but with some reasonable assumptions, I think you could fit that line also. I think so. No, here it's really artificially introduced by the experiment. But in general, in healthy tissue, so there's a very nice Nature paper in like 2020 or 2019, I don't remember. So there's something, I'm not a molecular biologist, so don't kill me if this is completely wrong, but there's something called the STING process or STING pathway. So what happens if you have chromosomal instability and you form these ring-like structures is a natural process. So DNA is bound to do that. So it's a natural process in your cell. And the cell recognizes that you have the circular structures and enter senescence. So it's actually a protection mechanism against. So if these pathways work, a cell kills itself if it has this ring-like structure. And obviously, like in cancer, things go wrong. Yes, right. And obviously in cancer, these things go wrong and they don't do that. But obviously, like all the cell lines that are used here, they don't kill themselves. But right. Yeah. Right. So that shows like if you follow it over time and you just amplify a random part of the ecDNA of extra chromosomally, it follows this neutral prediction. What about diversity? So before, I'm sorry that these equations are missing, but you don't miss important stuff. So the most important thing is here. before we asked what is the mean, what is the variance, and now if we take a tumor and we like segregate it in many, many single cells, and we ask what's the distribution of copy numbers in these single cells, right? So how many cells carry 1 copy, how many cells carry 50 copies, how many cells carry 100 copies. And you can, of course, do this in stochastic simulations, and you get these distributions that are very wide. So you get a peak somewhere here, but you get this very big tail of like cells carrying 50 copies, 100 copies, very high copy numbers. And then you can go to data again, and you can ask how does these distributions look like, and how does this compare to these stochastic simulations. And again, these are these cell lines that I showed you before, where we had these single cell divisions, and now you let them expand to reasonably big population sizes, and you ask what is the single cell distribution, again measured like by FISH. So you take 200 or 300 cells, look through the microscope, count the numbers, and then compare to the stochastic simulations. And you find that these shapes are very well described by this very simple theory of like simply growing and randomly segregating, and then you produce this diversity level. So to summarize like these observations, so what we first did was test if ecDNA is under random segregation. So these were these experiments with this like single cell divisions counting copies of ecDNA. And then we asked, is there a way of distinguishing positive or neutral selection easily, like just looking at these ecDNA copies? And there are simple measures that do that. So like, for example, if you look at the fraction, you say neutrality fraction goes to 1 for cells without ecDNA, fraction goes to 0 for cells carrying ecDNA, which agreed with like what we see in the cell line experiments. If you look at the mean is increasing. That's also what we see in data. And in the end, there were like, Vinny doesn't show you all of these experiments, but there were several tests, and all of them, if you look in patients, suggest that the ecDNA you detect in patients is under quite strong positive selection. I on purpose didn't put any numbers on it, so to classify how much positive selection, it's a slightly more difficult question, but there's clear evidence that there are under positive selection. And I think that's mainly to the bias that like we are in this lucky situation, like if you think about like ecDNA evolution, that like if you're in a neutral process, they are naturally suppressed by this evolutionary process. Whereas this is not true if you look at point mutations, for example. Point mutations, we have tons and tons of neutral point mutations at high frequency. Simply they occurred early in this like evolutionary process, like what Julio talked about yesterday, for example. Okay. So this is a relatively new field from an evolutionary point of view. So we just started basically. So this is the starting point. There are many, many open questions. We are just listing very few. We just talked about the copy number distribution of ecDNA. Of course, the next very natural question is like, what is the mutation distribution on these copies of ecDNA, right? So if you have a cell that carries like 150 copies of EGFR concentrated in a single cell, and we know that like point mutations in EGFR are very important for resistance evolution, for example, of course, and we immediately want to know what's the distribution of these mutations within these copies of ecDNA to understand like how resistance, for example, can be pooled into single cells with this process, for example. That's what we discussed before, like is there reintegration of ecDNA into cancers, and how would that influence diversity, and how can we detect that by looking at these distributions, for example? Or do we need time series data or something like that? Treatment, how resistance behaves, like given these ecDNA distributions, how space plays a role, how to quantify actually selection strength, to put numbers on that, and many, many other questions. To do that, so some shameless self-advertisement. So Vinny and I will have exactly 4 postdoc positions that will open at some point this year, hopefully. So they will be advertised at some point. So if you know anyone that is interested, or you are interested, or you heard of anyone that is interested, drop us a mail and we can discuss this. Thanks a lot. So there are a lot of people that worked on the experimental side. These are Winnie's. Many of them here. Christo is here, Lisa is here, Magnus is here. My students, Alex is here, Francesco is here. So also talk to them during the meeting. Thanks.